Thank you to the organizers for allowing me to participate, even though I can't be there in person. It's too bad. I would love to be there. And what I'm going to talk about today are some ideas around embodiments of diverse kinds of minds, and maybe the implications for consciousness. I myself do not work specifically on consciousness, but many of the things we do, I think, have implications for it. So today I'm going to go out on a few limbs and try to introduce some data to you that may be relevant. All the primary papers, the software, the data sets, everything is downloadable here at this laboratory website. And this is my own personal blog where I talk about what I think some of these things actually mean. So what I want to be clear today, especially for this audience, is that I do not claim to have a new theory of consciousness, nor am I going to try to support one specific theory of consciousness by data. But here are the things that I am going to claim. First of all, that the use of cognitive frameworks, in other words, mentalistic terms and all of the tools, both practical and conceptual, of cognitive and behavioral science, are highly applicable outside the brain. I do not think neuroscience is about neurons at all, and I think a lot of what we have gained from behavioral and neuroscience can be used far outside the brain. And this is not a linguistic, philosophical, or poetic kind of claim. Over the last 25 years, being able to do this has led to many new discoveries and new capabilities, both in our lab and in other labs. And so I claim this, the defense for this kind of weird idea is simply empirical fertility. I think that if we use any of the following criteria for addressing the problem of other minds with each other, so whether you like cellular mechanisms that underlie cognition in the brain, whether you like problem-solving behavior or evolutionary relationships, or even metrics such as phi and causal emergence and things like that, if you use any of those things as evidence of consciousness in addressing the problem of other minds, then for those exact same reasons, you need to consider very seriously the possibility of consciousness in many body structures. I personally actually would go significantly farther than that, but for today we're going to focus on biologicals mostly, with a little bit at the end of some other things. But I think those criteria are telling us that we have to take this very seriously. And I think that outstanding problems of AI and ethics and many other things are not going to be resolved if we insist on maintaining a restricted focus on humans, on three-dimensional space as the only kind of arena for embodiment, and on brains as privileged. I think all three of those things have to be widened quite significantly. So what I'm going to do today comes in four parts, a little bit about my framework, and then I'm going to show you what I think of as a really interesting model system for these kinds of studies, which is the collective intelligence of morphogenesis. We'll talk a little bit about some new creatures that have never existed on Earth before, and then I'll make a couple of conclusions and maybe say some fairly outlandish things. And so we'll see. The first thing I want to point out is that I think a lot of discussion in the philosophy of mind and in this field in general has been driven by this kind of picture. This is a famous piece of art called "Adam Names the Animals" in the Garden of Eden. And there's a couple of things here that I think are deeply wrong with this picture, but also something that's very, very deep and interesting. The thing that's wrong with it is that it gives this idea, which I think a lot of people have internalized, that there are discrete natural kinds. There are different kinds of animals. We know the differences between them. And so here's Adam. He's, of course, different from all the others. And I think this needs to go for reasons that I'll show you. But what I think is really important here is that in this ancient biblical story, it was on Adam to name the animals. God couldn't do it. The angels couldn't do it. It had to be Adam that names the animals. And in these kinds of mystical traditions, naming something means discovering or even somehow bringing forth the true inner nature of something. By giving it a name or by discovering the name, it means you've understood its true inner nature. And this I think is profound, because we are going to have to understand the inner nature of a number of other minds that are very different than ours. So the first thing I would like to claim is that ever since an understanding of evolution and an understanding of embryonic development, meaning that we arise through a set of slow and gradual changes from a single cell on these timescales, I would claim that the continuity thesis should be the null hypothesis. That is, it's not the case when I say these categories that we all like to think about in terms of conscious and the cognitive and so on. When people say, why do you think that they are continuous as opposed to categorical, binary sorts of things? I think it's actually on the onus is on those who do think that these are sharp transitions and categories to show why that makes sense. I think the background assumption should be that it's continuous and gradual. And if you think there are phase transitions, you have to say what those are and how they exist. So I take continuity to be the null hypothesis for all of this. Now, we are at the center of not just this continuum, but actually another continuum where both through biological changes and technological changes, the notion of a human, which figures very prominently in discussions of philosophy of mind and so on, which they typically mean the mature, sort of modern adult human here with his agential glow of responsibility and moral worth and all of that. Again, I think this is all a spectrum because it is no longer going to work to talk about humans versus machines or living things versus machines, because all of this forms a very tight continuous spectrum. And when we have things like this, it is not going to be feasible to start issuing some sort of proof of humanity certificates to figure out if he's got more or less than 50% of engineered components in here, because life is highly interoperable at every level of organization. You can introduce quote unquote machine like parts, and so this is another distinction that I think is going to disappear. So I've been working on a framework whose job it is to enable us to recognize, create, and ethically relate to truly diverse intelligences. And this means not just the primates and birds and things that, and maybe a whale or an octopus are the kinds of things that people often think about, but also on the exact same scale, I want to be thinking about all kinds of weird creatures such as colonies and swarms, synthetic engineered new life forms, AIs, whether purely software or with some sort of robotic embodiment, potentially someday exobiological agents, and some very exotic things that I won't have time to talk about today, including patterns within media and some even stranger things that I've talked about in other places. So what I'd like to do is to develop a framework that allows us to think about what do all of these things have in common. And very, very critically, it has to move experimental work forward. It has to give us new capabilities at the bench, and it has to enable better ethical frameworks. And of course, I'm not the first person to try for something like this. So here's Rosenbluth, Wiener, and Bigelow, who tried kind of a cybernetic scale all the way from passive matter to human metacognition. But this is the sort of thing that I've been trying to flesh out with newer data that these guys didn't have access to, and all the details are here if you want to see all the details. They're in this paper. So the first thing to note. So let's just talk about the biological material that underlies examples of cognition and consciousness of which we are aware. This is where it comes from. We all start life as a single cell. This thing, people with the unfertilized oocyte that people will look at and say, okay, well, this is subject to the laws of physics and chemistry, and then gradually and slowly it becomes something that is subject to the rules of behavioral science or even psychoanalysis and things like that. So one thing developmental biology tells you quite clearly is that there is no magical place to draw a bright line where you can say, okay, you were physics before, but now you're a mind. Okay, and so what we actually need are models of scaling and transformation. What happened to the competencies of this material in order to get to this kind of thing? And the question should be what kind and how much of cognition does it have? Not binary categories, is it or isn't it? And actually, there's a few interesting things that can happen even after that we can talk about. Okay, but at least some people will say, okay, sure, you can talk about ants and beehives as some sort of a collective intelligence, but maybe that's just metaphorical talk. Surely we are a true unified intelligence. We have this nice centralized brain, and we are, the human experience of being a unified mind is underwritten by this kind of singular organ. And so, for example, Descartes really liked the pineal gland because there's only one of them, and he felt that was a good place for the unified human experience. But he didn't have access to good microscopy. If he had looked in the pineal gland, he would have found all of these things, so tons of cells. And inside each one of those things, he would have found all of this stuff. And so nothing is unitary. We are all made of parts. And in fact, I would claim that all intelligence is collective intelligence. I'm not aware of any agent that is simply not divisible into other parts, that does what it needs to do sort of in some sort of monadic form. And so this is the kind of thing that we are all made of. So this is a free-living lacrimaria, but you get the idea. It's a single cell, no brain, no nervous system, handles all of its needs in ways that make soft matter physicists and roboticists very jealous. The degree of control it has over its body as it pursues its tiny local little goals. And if we want to say things about our own consciousness and that of even weirder kinds of novel engineered constructs, we need to be able to say something about this kind of creature and what happened to it when it joined into networks to enable it to become the kind of thing that we are. And Nick Rouleau and I try to make those questions very sharp in the form of a flowchart in this paper. Now, the interesting thing is that while this little guy has all kinds of interesting behavioral properties, in fact, it is even made of material that already has some of those properties. So, for example, it turns out that molecular networks, never mind cells, the nucleus, synapses, never mind any of that, just molecular networks, so represented by a small set of ordinary differential equations that describe how each chemical turns other chemicals on and off, so it's just a little network. Even these things are capable of 6 different kinds of learning, including Pavlovian conditioning. And we are using this, we're taking advantage of this in the lab for all kinds of medical purposes, such as drug conditioning and so on. You can train the material inside the cells. So when we think about ingredients to cognition and the kinds of things that go along with significant consciousness, such as memory and learning and so on, we have to understand this did not wait for brains and synapses to be formed. This was in many ways a free gift from mathematics, because the properties of these networks are actually to learn, and the ability to learn from experience in precisely ways that are recognized by behavioral science in terms of habituation, sensitization, anticipation, associative conditioning, all of those things. Those things emerge from the mathematics of these networks. They are not dependent on evolution of the substrate. In fact, something else that we discovered quite recently, and this paper is going to come out, I think, next week or something, that we actually found that if you compute causal emergence, and in particular, I know there's many metrics of it. We're using this one called phi-d. If you actually compute causal emergence of these networks during the training process, what you find, not in all of them, but in many of them, what you find is that actually the process of being trained causes their phi-d to go up. That is, instances of training and then testing actually reifies the network as more and more as being more than the sum of its parts. So this idea of being able to use causal emergence metrics to understand what kind of system, how much causality is in the higher level system versus its parts, again, that is not a brain specific thing. You can find these kind of phenomena very early on, and I could say much more about this, but I need to move on. But one of the most important things to realize about these kinds of systems is that they remind us that as humans, given our own evolutionary history, we are obsessed with the three-dimensional world. In other words, when we say behavior, or more importantly, when we say embodiment, what we typically mean are medium-sized objects moving through three-dimensional space at medium speeds. Those are the things we are primed to recognize as intelligent. And this is a problem for this field, because we find it very difficult to visualize things that biology has been doing long before brains and motility came on the scene. Biology has been navigating all kinds of other problem spaces. So what I think is the most important thing about quote-unquote embodiment, I think embodiment is really critical, but embodiment is not what we think it is. It is not the ability to, not just the ability to move around through three-dimensional space. Because this kind of loop, this perception, decision, action loop, happens in all kinds of spaces. Living things navigate the space of gene expression states. They navigate the space of physiological states. And what we'll talk about the most today is anatomical morphospace. They navigate the space of possible shapes. And in all of these cases, it turns out you can use the tools of cognitive and behavioral science to understand how they navigate these spaces and the degree of ingenuity they deploy. And I think that if we had some sort of inner sense where we could directly sense the way we do with taste and so on, either gene expression states or physiological parameters in our blood, I think we would have no trouble realizing that our liver and our kidneys are these amazing symbionts that intelligently navigate these other spaces to keep us alive every day. So we need to widen this notion of what embodiment really means and be broader about what kind of spaces intelligence exerts itself in. Now, the last thing I'll say about my sort of philosophical cards on the table is that what I think cognitive claims are basically interaction protocol claims. In other words, when somebody says this system has a particular degree of cognition, intelligence, whatever, I don't think we're talking about what it really is in its essence, as some sort of ground truth that we can argue about for a bit and then eventually settle on the correct answer. I don't think that's what's going on here. I think what you're really saying when you make those claims is that here is a set of tools that I bring to interacting with the system, and those can be hardware modification. It might be cybernetics and control theory. It might be behavioral science. It might be psychoanalysis and love and friendship and whatever happens at this end. But what you're saying is I'm going to bring this bag of tools, and then I'm going to interact with the system, and then all of us, as a matter of empirical discovery, can find out how well that worked out for me. So if you assume that cells and tissues, as modern molecular medicine does, are somewhere down here, they're mechanical machines that need to be rewired with genomic editing and things like this, you're going to leave a lot on the table, because actually testing the applicability of some of these tools makes many discoveries that are not accessible at this level. So I think I think all of these things are empirical questions. I don't think they can be settled from a philosophical armchair. We cannot just decide. We should not be tied to categories, ancient philosophical categories. Those kinds of things have to advance with the science. And the question simply is this: if we assume a continuum hypothesis, then we are open to testing tools from other disciplines, typically reserved for minds and especially humans in other cases, and see what happens and see what kind of empirical purchase we get on this. Okay, so at this point, I'm going to talk about some very specific examples using a model system. The model system is the collective intelligence of cells making decisions, learning, and meeting goals and failing to meet other goals in the process we call morphogenesis. Now, I preface this by saying that actually, I think Turing was onto this. I think he didn't write about this directly, to my knowledge, but I think he saw clearly a deep symmetry between the self-assembly of bodies and the self-assembly of minds, because he not only talked a lot about different embodiments of mind and reprogrammability and intelligence in diverse implementations and so on, but he also wrote this really interesting paper on the chemical basis of morphogenesis. And why would somebody interested in intelligence be looking at chemicals in early development? Because I think he realized this is fundamentally the same problem, and there's something interesting. I used to have my students do this by hand, and then we developed a tool to do this that you can all play with. The game is this: you take a paper in neuroscience, you throw it into Microsoft Word, and you just do a find replace. Anywhere it says neuron, you say cell. Anywhere it says milliseconds, you say minutes. Anywhere it says something about three-dimensional space, you say anatomical morphospace, and that's it. And you usually end up with a very interesting developmental biology paper. The symmetries are quite striking, and you can try this yourself, and it's described here. So what I'm going to talk about next is to give you some specific examples and then show you some discoveries that this idea has led to. So the first idea is that morphogenesis is not hardwired. It is simply not the case that you start with an egg, then eventually then you just follow some rules that the DNA laid out, and then by emergence you get some kind of complex thing, this sort of feed-forward process. That is not actually what happens. What does happen is a goal-directed navigation of problem space. So here's an example. This is an axolotl, and axolotls are very good at anatomical homeostasis, which means that if you amputate some part of their body, for example the limb, you can amputate anywhere you want. The cells immediately can detect that they've been deviated from their correct position in anatomical space, and anatomical space has many dimensions. I can't really draw it, but you get the idea. The cells will very quickly try to reduce that error, and then the most amazing thing about regeneration happens. Then they stop. How do they know when to stop? Well, they stop when the correct structure has been completed. Now, this is an example of anatomical homeostasis. The system tries to reduce the delta between where it is now and where it wants to be. This means that it has to store a set point, which I'll show you exactly how that's stored. So it has to store a memory of what is the correct shape. And the other thing is that you can do this again and again, and it will keep doing it. About 5 or 6 times, actually. Eventually, it stops. It just stops. And whether that's a true instance of learning, we don't know yet. But it may well be that eventually it just gives up and no longer wants to do this anymore. But one important thing is that this whole process of anatomical homeostasis is not about repairing damage. It's actually much more interesting than that. If we take a tail, and this is old work from the 50s, if you take a tail and surgically attach it to the flank here of the salamander, let's say between the two limbs, what will happen is that over time it will get remodeled into a limb. Now think about what that means. The cells sitting up here at the tip of the tail are fine. They're tail tip cells sitting at the end of the tail. There's nothing locally wrong with them, but they turn into fingers. Because what the system is doing is not only does it have a stored set point of what a correct layout should be, it should have a limb in the middle, not a tail, but it's got this interesting kind of top-down causation where all the molecular steps that need to happen in order to turn tail structures into limb structures are in the service of a larger scale goal, of which they probably know nothing about, but the system as a whole filters down this very high-level design spec of having a limb instead of a tail, filters that down to the behavior of cells and molecules, exactly as happens in cognitive systems, where your very abstract social and financial goals have to be actually transduced down into moving ions across your muscle membrane so that you can actually move around and execute on those goals. So this idea of local order obeying a global plan is very interesting. There are, if you ask why the cells are doing what they do, you run into the same kind of thing as with explaining instances of behavior. You can try to give molecular explanations. Well, it's because this molecule signaled to that receptor and so on. But actually, that's not the deeper answer as to why it's happening. The real answer as to why it's happening has to do with the target state that it's trying to achieve. And actually, to take it even one step further, to link it further to a problem solving and intelligence, that kind of remodeling has some amazing examples. This is one of my favorites. This is a cross section through a kidney tubule. And what you'll see 8 to 10 cells are making this kind of structure. One thing you can do is make polyploid newts that have extra copies of their chromosome. If you do that, the cells get bigger. As the cells get bigger, you find that the amazing thing is that it still makes an animal of exactly the same size. Well, how can that be? Because it's scaled the number of cells to the new cell size. And if you make truly gigantic cells, and these are like 6N polyploid newts, only one cell will bend around itself, abandoning the molecular mechanisms used here, cell-to-cell communication, and use cytoskeletal bending to bend around itself to solve the problem and create the same structure. So that same journey in anatomical space, the system is using different tools in its genetically specified set of affordances to get to where it needs to go. Now, this is a standard component of many IQ tests. Here's a bunch of objects that you have, used them to solve this problem you haven't seen before. And so think about the plasticity here. If you're a newt coming into the world, never mind uncertainty about your environment, you can't even trust your own parts. You don't know how many copies of your genetic material you're going to have. You don't know how big your cells are going to be. You have to use whatever tools you have creatively to solve the problem that you have, which is to get from point A to point B, from where you are in the anatomical space as an egg to where you're going to be as a good newt. And there are many examples. We could do this for hours. There are just remarkable examples of this. So actually, morphogenesis does exactly the kind of creative confabulation that I think goes on in cognition, where at any given time slice, as an agent doesn't have access to the past. What you have access to are the memory engrams, the messages left by your past self. And at that point, you have to interpret, like all messages, your memories have to be interpreted. They don't need to be taken literally, they need to be interpreted. And there's some examples that show you that they can't be taken literally. For example, memories that pass from a caterpillar to butterfly and they get remapped into a novel embodiment. And this process of continuously reinterpreting and telling the most adaptive, forward-looking story you can is exactly the same for cognition and for morphogenesis. morphogenesis, where you have this now moment, and everything that you've experienced before, be it behavioral memories or genetic information from your lineage, is squeezed down into very sparse representation, the genome or memory engrams, and then has to be creatively decoded. And all of this this is described here. So these parallels are really deep, I think. And in particular, this idea that morphogenesis, just like behavior, is a kind of creative storytelling of using whatever information you have, gives biology amazing plasticity. Just like for behavior, morphogenesis has it too. Here's an example. Here's a tadpole. So here's the mouth. Here are the nostrils, here's the brain, the gut. And what you'll notice is that we prevented the primary eyes from forming, but we put an eye on its tail. And I'll show you in a minute how we do that. Turns out these animals can see perfectly well. How do we know? We built a device that trains them on visual cues. And it turns out that, so these eyes form. They make an optic nerve. It does not go to the brain. It goes sometimes to the spinal cord, sometimes to the gut, sometimes nowhere at all. And these animals can see. It does not take new rounds of mutation, selection, adaptation. This works out of the box. You've changed drastically the sensory motor architecture of this animal. No problem. It works right out of the box. And I think that's because of this incredible it's because of this incredible plasticity that the tissue has, that it interprets its local situation to tell the best story that it can. It is not committed to whatever its ancestors were doing to with that information. And so before I show you how all of this works, I just want to give you one simple example to remind us of the idea of the importance of storing pattern memories. And that this is something we discovered a few years ago, that these tadpoles, in order to become a frog, they have to rearrange their face. So this is metamorphosis. The eyes have to move, the nostrils, the mouth, everything has to move. And the standard assumption in the field was that this is a hardwired process, that the genetics somehow tells every structure which way to go and how much. Well, as I said, this is you can't just assume these things. You have to do perturbative experiments to find out how much competency the system has at problem solving. And so what did we do? We created these so-called Picasso tadpoles. So they're scrambled. We scrambled all the craniofacial organs. The eyes on top of the head. The mouth is off to the side. The whole, it's like a Mr. Potato Head after you scramble all the parts. Well, it turns out that those tadpoles make largely perfectly normal frogs, because the genetics does not give you a hardwired set of rearrangements. What it actually gives you is a system that can execute error minimization, and all of these things will move through novel paths to get to where they're going, despite the fact that they're starting off from weird positions, and actually sometimes they go too far and they have to come back. So this tells us several things. The system has to have a mechanism for knowing what is the correct position. How do you know you've gotten to the right place? And it has to have mechanisms for actually computing the means ends analysis of how do I reduce error? What am I going to do to reduce this error? And so well over 25 years ago now, what I did was to simply say, okay, well, what is the one example that we know of where groups of cells store goal states and try to execute towards those goal states? And that, of course, is the brain. And so I don't need to, in this audience, I don't need to go into all of this, but just to say that there's ion channel and gap junction hardware that underlies this electrical network, of course, also neurotransmitters and many other things. And there's this process of project of neural decoding, this idea that if we could just read out the electrophysiology, and if we knew how to decode it, we would be able to read out the cognitive content, the mental content of the animal. We would know the goals, the preferences, the competencies, if only we knew how to read out this kind of electrical activity. So it turns out that, and probably shouldn't have been too much of a surprise looking at it now, this idea that actually the ability of this kind of hardware to do these amazing things is absolutely ancient. Evolution discovered the benefits of electrical networks for integrating and processing information long before neurons, in fact, long before multicellularity showed up. Around the time of bacterial biofilms, this was already, bioelectric networks were already being used to create proto-individuals that were more than the sum of their parts. And every cell in your body has ion channels. Most cells have these electrical synapses known as gap junctions, lots of neurotransmitters being part of this network outside the brain. And so this is critical to my claim that if you think consciousness is associated with brains because of the amazing hardware that the brain has, That hardware is ancient. It long predates neurons and brains, and it is present throughout biological bodies. And so that raises the interesting question. We know what these networks think about. What did these networks think about before there were brains and before there's a brain in the embryo? What do they think about? And so basically we undertook the process of stealing as many tools and concepts from neuroscience to ask very similar questions. Could we do a kind of decoding here to ask in these embryonic examples what are these networks doing? So we developed a number of tools. First of all, voltage-sensitive fluorescent dyes and genetically encoded reporters to be able to, and these are not models. This is real data. So this is an early frog embryo, and these are some explanted cells. To look at be able to read out all the different voltage patterns and all the conversations that these cells are having with each other, and then use all kinds of modeling integrated with the molecular biology of what ion channels underlie these gradients, and then of course tissue level dynamics of what's happening with the voltage gradients as a function of time, and then continue scaling up so that we can integrate that with some ideas in dynamical systems theory and connectionist ideas around networks that store memories and can do pattern completion, right? So that regenerating a limb that I showed you is an example of pattern completion, and so we can sort of integrate across the levels of organization here. Okay, but that's just reading the patterns. More importantly than that, you want to be able to write patterns, because it's not enough to just do observations. You have to do perturbative experiments. So how do we communicate with? And I really think that what we're doing here, and I'm about to show you some examples of this, is not simply trying to micromanage physiological states on some kind of machine. I think we're actually communicating with an unconventional decision-making, memory-forming intelligence here. And in order to do that, we use, again, not surprisingly, the electrical interface. In other words, no magnets, no electromagnetics, no applied fields, no frequencies. What we do is we hack the interface that the cells use to become a larger level entity. In other words, the cognitive glue. This just like in our brain, the electrophysiological processes are the cognitive glue that make us more than a pile of neurons. And so what we can do is we can open and close these channels, we can open and close these gap junctions, and so we can manipulate the synaptic and the intrinsic plasticity of any tissues in the body. And then we can ask what happens when we write new memories, new voltage patterns into the system. So I'll show you one of my favorite examples. We have a particular voltage state that we can induce by injecting RNA encoding certain ion channels. If we inject those RNA into the early embryo, this is an early frog embryo, into a bunch of cells that are going to become this gut region, what we can do is set up an eye-specific voltage pattern, and sure enough, those cells get the message, they make an eye. If you section those eyes, you get the same lens, retina, optic nerve, all the stuff that eyes are supposed to have, so it's a proper eye. And this teaches us a few things. First of all, that these bioelectric patterns that we're putting in are instructive for behavior. In other words, we can communicate a message that will guide the way that the collective navigates morphospace. Again, remember the claim. The claim is that groups of cells are a collective intelligence. They use electrical networks, just like our brains do, to be the cognitive glue of that larger scale intelligence. And the decisions they make are about movements in anatomical space. And so here we can provide a message that causes them to go somewhere else and build an eye instead of a gut. It also tells us that, much like when I'm communicating with you now, I don't need to worry about adjusting the synaptic proteins in your brain. I'm giving you a very thin information signal through language, and then I rely on you to adjust the molecular biology and biochemistry in your brain to make it make sense. Same thing here. We're not communicating with the genes. We're not telling the individual stem cells what to do. We're providing a very high-level, abstract signal that means it's actually an arbitrary symbol that means because that's how the cells interpret it. It means make an eye here. That is all that we said. The rest, the material takes care of the rest. And then I want to show you something interesting that happens here, which again I think has interesting parallels to what happens in the brain. If we take a cross section through one of these eyes, what you'll see is that the blue cells are the ones that we injected. But all this other stuff, we never touched it. Why are they participating? And so what actually is happening here is that the cells that we inject are recruiting their neighbor cells the way that other collective intelligences like ants and termites recruit their nest mates when they find something too heavy to lift. They recruit them to build this eye. And we didn't teach them to do that. Of course, they already know how to do that. But here's something interesting that happens. If we do this, we inject this message, and then we look in the early embryo. This is a much earlier stage embryo than this one. What you see 4 or 5 nascent ectopic eye fields. These are the cells that got the message and are just starting to sort of flirt with the idea of making an eye. They're starting to express early eye markers like RIX1. But when you let this embryo grow up, one of the things you'll notice is that almost always most of these resolve and don't actually become eye. Only some of them do. And that's because what's actually going on here is a battle of patterns for the future. These cells, the ones we injected, are saying to their neighbors, you should be an eye with us. You should help us become an eye. But there's a cancer suppression mechanism at play here where the cells with abnormal neighbors, the first thing they do is try to tell them to normalize and be like them in terms of voltage. And this is a whole thing I won't talk about, but we have a whole oncology program centered around that mechanism where the local cells are saying, no, you shouldn't be an eye, you should be skin or gut or whatever it is, like us. And what you have there is a battle of patterns of who is going to dominate the future. Which pattern? Are we really going to give in to this new message and become an eye, or are we going to stick with this with our priors and be a skin or gut? And so we can actually watch this happen. Now here I want to show you what it looks like at the single cell level for a convincing message to be passed along from one subunit to another. So I'm going to play this video in a moment. What you're going to see is this cell, which migrates around here. You can see that the voltage, the color here indicates the voltage. So the voltage here before it encounters this whole mass is different than the rest. So it has a more depolarized voltage. But as soon as it touches, and this is all it takes, is a tiny little touch. As soon as it touches, it feels the voltage of this cell, and that voltage takes over. The cell basically becomes hacked by the bioelectric information that is present in this collective. And after that, it becomes the same, and you'll see it joins. So here it is. Yeah, the colors are unfortunately inverted here, but you can see it's quite different than the rest. Bang, there it is. It touches the rest, and now it's got the same voltage, and now it merges with this collective. So what you're seeing here is a very convincing bioelectric state taking over this subunit, which then joins a collective with a larger cognitive light cone. This network has greater capacity to pursue morphogenetic goals than single cells do. And so this is what it looks like at a single cell level. But these kind of pattern memories, these bioelectric pattern memories really come into their own in the multicellular context. And what I want to show you is how we read and then rewrite the goal states, the anatomical set points for this tissue, again, as an example of a collective intelligence. So the whole point of collective intelligence is that you make individuals that are composed of parts where the higher level individual knows things and can do things and can pursue goals, larger goals than any of the parts can. And so what we see here is an experiment, a set of experiments that we did over about a decade with these flatworms. These are called planaria, and they have a number of amazing features, but one of the features is that if you cut them into pieces, each piece regenerates exactly what's missing, and then it stops. In other words, every fragment of this body knows exactly what a correct planarian is supposed to look like, and it does what it needs to do to reduce the delta between the injured state and the final goal state, and then it stops. So what you see here is that a single normal planarian, it will have a molecular marker expression of anterior state up at the head, not at the tail. And when you amputate the head and the tail, the middle fragment extremely reliably generates a worm like this: one head, one tail. So this happens basically every single time correctly. And one wonders, how does it know how many heads to have and where to put those heads? Because this piece grows ahead. This region of this piece here grows ahead, but so does this one back here. The anterior portion of the tail is also going to grow ahead, but the posterior portion of the mid fragment is not. It's going to grow a tail, and yet all of these cells were direct neighbors to each other, meaning that the positional information is the same, but the fates are quite different. The anatomical fates are quite different. So you see here, this decision is made holistically. It cannot be made locally based on just the position. It has to be made in the context of what else is here, which way is the wound facing, that kind of information. And so one way that these fragments know how many heads is that they have this bioelectrical pattern. And this is, again, voltage sensitive fluorescent dyes that we use to determine that there's one region here that says one head, and one tail. And so what we were able to do, and the technology is still pretty messy, it's still being worked out, but what we were able to do is to change that pattern by exposing these animals to ionophores, which allow specific ions to enter the cells, changing the voltage in a predictable way. And this, it only takes 3 hours. The regeneration process takes about 8 days. The voltage change happens in 3 hours, and I just showed you in a single cell it can happen very quickly, right? In this kind of context, it happens literally in a matter of minutes. You touch and that's it. But in this context, it takes about 3 hours minimum. But when you do this, we can establish a pattern that looks like this. And while this pattern is interpreted by the regenerating system as build one head, build one tail, this pattern is interpreted as build two heads. And so if we do that to this animal and then amputate the head and the tail, this middle fragment will build two heads. So this is not Photoshop or AI or anything like that. These are real animals. And what's really critical here is that this bioelectric pattern is not a map of this two-headed animal. Okay, you see two-headed bioelectric state, but that is not a map of this animal. It is a map of this perfectly normal, anatomically normal planarian. It has one head, one tail. If you look at the molecular markers, there they are, anterior markers in the head, no anterior gene expression in the tail. So what you're looking at here is a latent memory. This is a bioelectrical memory of what a correct planarian is supposed to look like, but it's latent. The tissue doesn't do anything with it until it gets injured. Only if it gets injured does it consult this internal map of the goal state, and then it builds what it needs to build here. And so you can think of this as a primitive sort of evolutionary ancestor of the brain's amazing ability to do mental time travel. That is to imagine and remember circumstances that are not true right now. So again, this is a counterfactual memory. It is what I would do if I got injured in the future. And I think, again, this is telling us that these same exact mechanisms that are exploited by brains and nervous systems to do memory and prediction, we're already doing this kind of thing, but in the anatomical space, long before brainy kinds of behaviors in three-dimensional space. So again, yeah, this is how we read and rewrite the content of the mind of the collective intelligence, which is using these patterns to help it navigate anatomical space. Now, I keep calling it a memory, and one way to show that it really is a memory is to point out that bioelectrical state, once we induce it, it holds. It's permanent. So when we take these two-headed animals and we cut them again and again and again, as far as we can tell, indefinitely, no more exposure to any drugs, just in plain water. We keep cutting it, or in fact, we let them fission on their own. The normal reproductive mode of this animal is they tear themselves in half, and that's fine. This propagates. Basically, once you're two-headed, the fragments will be two-headed in perpetuity. Here you can see, by the way, these animals moving around. The first time I showed this at a conference somewhere, somebody stood up and said that those animals can't exist. And so I make sure to bring these videos so you can see they absolutely do exist. And these are from the third or fourth generation, and you can keep doing this. It's permanent, because these until we set it back, and we can reverse the bioelectric memory and set them back to one-headed. But this has all the properties of memory. It's long-term stable, but it's rewritable, so it has some lability to it. Once we know how to do it, we can rewrite it. It has latency or conditional recall, meaning that these memories can be latent and not expressed until certain conditions are met. For example, injury stimulus triggers the recall of that memory, and there are some discrete behaviors: one head, two head, and so on. However, it should be pointed out that this system is not just for head number, and while it is the case that a single planarian body here can store at least two different internal representations of what a correct planarian looks like, probably more, but we've nailed down these two. It's actually not about head number at all. There's much more here. For example, head shape. So we can take a planarian that has this nice triangular head shape and cut off the head, perturb the bioelectrical network that allows it to remember what kind of head it's supposed to make. And then you can get things like this. You can get this flat head like a P. felina. You can get round heads like an S. mediterranea. Of course, you can also get the normal heads. And so, and not only the head shape, but the shape of the brain and the distribution of stem cells becomes just like these other species. About 100 to 150 million years of evolution between these guys and this one. And remember, there is no genetic change anywhere here. There's no genetic change in the two-headed worms. There's no genetic change here. much like every cognitive system that we are familiar with, the whole point is to learn without needing to radically change the hardware. You don't need to mutate your DNA to learn new things or to express the memories that you have. And the same thing is true here. Normally, this hardware finds one very particular attractor in the anatomical morphospace. So here's where they live, right here. And however, it is perfectly willing to visit other regions here if it gets confused. And the way it gets confused is the bioelectrics that help it navigate that space towards a specific location. We now have the ability to alter those, and then they will end up in these other some of these other attractors. So you can see again how they're navigating anatomical morphospace using some of the same mechanisms that that we see in the brain. Okay, so here basically we come to this next to last section, because what I've shown you up until now are endogenous examples, meaning natural, evolved, or early ways that biology uses representation, memory, and the bioelectrical interface to store goal-directed set points that then, with various degrees of ingenuity, the collective intelligence of cells can follow. And what we want to do now is ask the following question: in these natural examples, when one asks where do the goal states come from, usually we say, well, it comes from a combination of heredity, so a long history of selection for a particular pattern memory with certain environments, and there are some things that are constraints of physics and so on. But what we wanted to ask is more broadly, in novel cognitive systems, where do goals and memories come from? That is, when we deploy the certain genetic hardware in a completely new lifestyle that has never faced selection in that configuration, what actually happens? And so here we turn to synthetic morphology, and I'm going to show you first this particular example. These are called xenobots, and the way they form is we take early frog embryos, and we take some of the epithelial cells at this early blastula stage from their the apical end of the embryo, and we dissociate them, and we put them in a dish. Now, there's many things they could have done. They could have died. They could have spread out and moved away from each other. They could have formed a nice two-dimensional monolayer like cell culture. But instead, what they do is they sort of coalesce into this little mass. We'll get to that flashing in a minute. It's the flashing is calcium signaling, but you can see these cells sort of coming together, and actually, if you look closer, there's some amazing things that they do when they do come together. And then the whole thing, because it is ciliated, it's covered in these cilia, whose job normally is to distribute mucus down the side of the animal. In this case, what it does is it rows against the water, so they're able to move. And so they can go in circles, they can patrol back and forth like this, they can have collective behavior. So here's two of them interacting. These are sort of resting in place. This one's going on some sort of very lengthy journey. So all kinds of behaviors. You can see it here traversing this maze. The still water. The water's not moving, and also there's no gradients. There's nothing in there besides water. You can see what happens. It takes the corner without having to bump into the opposite wall. So here it comes. It's moving along straight, and without having to hit anything, it takes the corner. And then here, spontaneously, it turns around and starts to go back where it came from. Okay, so all of this is completely internally driven. It's autonomous. We're not pacing this. We're not controlling the movement the way that the people do with some engineered biobots. This is completely autonomous, and they have all kinds of unusual behaviors that were not predicted in advance. For example, one of the things that it turns out they can do, and this was this actually was found in some it was found in some simulations that our collaborator Josh Bongard and Sam Kriegman did, that basically if you provide loose epithelial cells to the bots, what they'll do both on a collective population level and on individual level, they will sort of polish them into these balls. They collect them and polish them. They collect them into little piles, and then they polish them into these little balls. And because they're dealing with an agential material themselves, in other words, these are not passive particles, these are cells, these balls then mature and become the next generation of xenobots. And guess what they do? They run around and do exactly the same thing, and they make the next generation and the next. So we call this kinematic self-replication. It's kind of von Neumann's dream of a robot that can go out and make copies of itself from material it finds in the environment. As far as we know, no other creature on Earth does or had reproduced by kinematic replication. This is entirely new. And so when we make it impossible for them to reproduce in the normal frog-like fashion, they are able to do this. And so that leads to, again, these questions of plasticity. That what did the Xenopus genome learn in all of its time on Earth? Well, it certainly can do this, and this is what it does very reliably under standard circumstances. This is the default behavior of that hardware. You can think about this as like instincts, the way that this stereotypical morphogenesis is the way that like some birds are born knowing how to make certain nests and things like that. So this is the kind of instinctual built-in behavior, but there's much more plasticity. And when these cells are liberated from the embryo, remember, we didn't add anything. There are no the nanomaterials here, no scaffolds, no genomic editing, no synthetic biology circuits. We did almost nothing to them except liberate them from these other cells, which normally bully them into having a boring life as the two-dimensional outer covering of a frog that will keep out bacteria and things like that. In the absence of those signals, you get to find out what these cells really want to do. And what they want to do when shielded from the instructive influence of other cells is this. They become xenobots. This is I think an 84-day-old xenobot. It's turning into something. We have no idea what this is. So it's got some sort of weird developmental sequence of its own. It has different behaviors than these tadpoles do. And probably this is just the tip of the iceberg. My guess is these things can do many different things under many different circumstances. So it's very interesting because there has been no selection to be a good xenobot. There's never been any xenobots. There's never been any selection to do kinematic self-replication or to have these kind of patterns and behaviors and so on. So that really leads us to ask, as a model system for what I'm going to talk about momentarily, which is the kind of diversity of embodied minds that we're going to be dealing with. This is a model system for that. Now, I'm not yet making any claims about their level of intelligence, their preferences, their goals, or their consciousness. So some of this work is ongoing. We should probably have some things on this later this year, as far as what they can learn and so on. But certainly you can't say any of these things just from observing this kind of behavior. So you have to do perturbative experiments, which is what we're doing. But one of the interesting things we found is that if we look at the gene expression in these xenobots, turns out that they express hundreds of genes differently than the age-matched tissue in their normal context. They turn on. We're not worried about genes that are not expressed because, of course, it's obvious xenobots are missing all kinds of tissues. They're missing endoderm and so on. So we're not worried about that. But much more interesting are what are the genes that they upregulate, and there are hundreds of new genes that they upregulate, which are very interesting. And again, we didn't touch the genome. We didn't give them any new synthetic biology circuits. So all of these gene inductions are because of their new lifestyle. They're dipping into the genome to express new genes that are relevant to their new lifestyle. And one of the clusters we identified is a set of genes that are known to be related to sensory perception of sound. And we thought that was really weird. Could it be that these xenobots are able to hear? And so we did this kind of experiment where we put speakers underneath the dish, and so what you can see here in this video is that this particular bot, for example, moves in a particular pattern. Then we turn on the sound, and so this is one particular frequency that we've been playing with. You turn on the sound, and you can see the behavior change radically. And as soon as you turn it off, it goes back to what it's doing. Not exactly the same, so there is a little bit of memory here, but it does return to its behavior. So we can in fact signal to them via sound and change their behavior in observable ways. So again, this question of what does the transcriptome look like of an entirely novel organism that's never existed before. This is it, and it has. I'm sure. Again, this was probably just the kind of one of many interesting things that we can discover about these. Now, here's something else that we're just beginning, and this will perhaps have even more relevance to to this audience who is interested in consciousness and various metrics thereof. So what we've got are xenobots that express calcium reporter proteins. So this is GCAMP that is flashing anywhere that you see calcium signaling. And of course, people use this in brains all the time. So neuroscientists use this exact construct to track computational activity in the brain. And then once you do that, you can apply a couple of different sets of metrics to them. So first, you can do various connectivity metrics, and this is a collaboration with Sarah Walker's lab and Josh Bongard's lab. and Olaf Sporns and all the people who worked on this, basically looking at the information structures and the connectivity that you find in these kinds of tissues and comparing them to what you find in nervous systems. So if you want to dig into those details, you can take a look here. I don't have time now to go into it. But I will just mention something else that's really interesting is that there are metrics that have been developed by a number of people having to do with integrated information, and the idea is to have some quantitative estimate of to what extent does the larger scale system have causality that is more than just the sum of its parts. Okay, just very brief kind of explanation, but I'm sure this audience knows all about it. So this is one particular metric. This is called phi r. Which is basically it's a measure of integrated information. It's based on Phi 2.0 by Balduzzi and Tononi, and so this was done by Thomas Varley in the Bongard lab, which they analyzed the Phi r of calcium signaling in these bots compared to null models, and they found that just like in fMRI, where you can see a clear difference of fMRI of brains. So you see this causal emergence is quite different in the brain versus the null model. The same thing is true here. Okay, so there is a considerable amount of integrated information. So I'm not going to make any claims about consciousness here. There are plenty of people who have discussed the relationship between consciousness and measures of integrated information and so on. So you can sort of interpret that how you want, except that if with just one thing, that if you like measures of integrated information applied to brains as a sign of some sort of integrated consciousness, you're going to need to deal with the same issue here, because even though there is no neural tissue here, you're finding the same kind of structure. So in terms of the signaling, so you can make of that what you will, and we have much more on this coming. But it's quite clear that these kind of metrics, which are being used in neuroscience to look for correlates, measurable correlates of consciousness and things like that, are very applicable to these kind of non-neural systems. Okay, so. In the last couple of minutes, I want to say some other things that might be in particular relevant to this particular audience, having to do with what I think this all means. So first of all, because of the plasticity of life, pretty much every combination of evolved material, engineered material, software, and the ingressing patterns of mathematics, which are in some sense not in the physical world, pretty much any combination of this is some sort of viable agent. So all the biologicals, all the endless forms most beautiful that Darwin was so impressed by, are like a tiny little corner of this enormous space of embodied minds. And so when people think about what's going on in whales and octopuses with their semi-autonomous limbs and some of those things, that's just a tiny piece of this whole question. I mean, hybrids, cyborgs, all kinds of chimeras, every possible version of this material is going to be living with us in the next few decades. And I think that having a theory of mind about these things, and a theory of consciousness that goes beyond evolution and beyond the N of one example that we have in the evolutionary tree here on Earth, and understanding it more broadly in very diverse embodiments that are very different from ours, is going to be essential to enabling us to reach some kind of ethical symbiosis with these beings with whom we're going to share our world. And just to say a couple of somewhat more wild things towards the end of this talk, since this is a meeting about consciousness, I want to say two things, and then if people want to dig in, we can discuss them. First of all, my view of where do the specific goals, competencies, and behavioral propensities come from if it's not a history of selection? So in novel beings, such as the bots that I showed you, of course, we also have anthrobots, which are biobots made of adult human cells, and all of the different cyborgs and hybrids and all of these different things. Where do their goals and competencies and kinds of minds come from if you can't pin it on a long history of selection? And all I will say here, because I'm running out of time, is basically that I think they come from the same place where the truths of mathematics come from. So Platonist mathematicians believe that they are exploring, not creating, but exploring an existing an ordered space of truths. These truths, the properties of prime numbers and the facts of number theory and all of these things. They are not determined by anything in the physical world. However, they matter greatly to physics, and I'm going to claim to biology as well. And they occupy an ordered, structured space. They're not merely a grab bag of emergent phenomena that just happen to hold in our world for some weird reason. A lot of people like that model better because it's sort of a sparse ontology. You don't need to think about a Platonic space of truths besides the physical space. But I think it's a very Mysterian position. I don't think it's helpful for future research to assume that these emergent things are just kind of randomly to be encountered and written down in our big book of surprises. I actually think the research program here is to map out this space, because this space, for lack of a better word, this Platonic space, contains not only these very low agency kinds of true facts of mathematics and so on. I think they also contain behavioral patterns that we would recognize as kinds of minds. Okay, and some of these patterns are behavioral patterns of cells and tissues, meaning they come out as morphologies or anatomies, and other patterns are ones that drive behavior in the three-dimensional world, which we recognize as conventional behavior. So I think that all bodies, be they simple machines, embryos, biobots, AIs, humans, embryos, whatever. All of these things are basically interfaces. They are pointers there are pointers through which some very specific patterns ingress into the physical world from a kind of Platonic space, and that understanding this physical interface is only the first part of what we need to do. We also need to understand what kinds of behavioral patterns affect and manifest through these physical interfaces, just as we try to understand how symmetries and truths about very abstract objects control various aspects of physics from the lowest level on up. And I think that synthetic morphology beings such as xenobots, anthrobots, and all of those things are basically vehicles for exploring this latent space. And I think they are an indispensable tool for consciousness research as we try to understand what kinds of patterns are important for human consciousness, and what else is in this space. I think we're going to have to map out the space to have a proper understanding of the mind-brain relationship. And the final thing I want to say is this: it's often thought that there's some essential relationship between complexity and consciousness, that in order to have consciousness, you need to reach a certain level of complexity of organization, and whether it's emergent or, as I think, it's actually a view from this Platonic space of kinds of minds. Either way, it's very common to think that not only do you need biology for that to happen, but you actually need very complex biology, for example, brains. And we are looking at some extremely minimal systems. I will just, again, running out of time, so I'm only going to just point people to this one paper where we studied sorting algorithms like bubble sort and things like that. And these things are very simple algorithms, completely transparent, deterministic. People have been studying them for many decades, and yet they have features that have never been noticed before. What we found is that they have all kinds of not only interesting competencies that are not obvious from the algorithm at all, such as delayed gratification and other kinds of behavioral tricks, but they also do these weird side quests in addition to the sorting that they do. So this is the sorting of the random string of numbers, and certainly they do the sorting. But in the meantime, they do this other interesting thing called clustering, which is it's nowhere in the algorithm. It is something that is not forbidden by the physics of their world, but neither is it prescribed by it. So it's an interesting new kind of freedom that is neither determined nor randomness. I think ultimately this kind of thing will have impact on kind of the free will problem and things like that. But basically, what we're finding, long story short, what we're finding is that even extremely simple systems that are not like biology whatsoever, that don't have a history of selection and so on, even very simple deterministic kinds of systems have behavioral competencies that are not directly encoded in the algorithm. You do not have to be a complex life form with a brain to do things that are that are not obvious from your construction. And I think that for the exact same reasons we understand that the laws of chemistry do not tell you everything you want to know about the human mind. For those exact same reasons, we should cultivate some humility and realize that the algorithm and the materials of which quote unquote machines, including very simple machines, are made, does not tell the whole story about them either. In other words, they too are subject to interesting ingressions from this Platonic space of forms that we did not see coming and that are not fully circumscribed by our stories of the material and the algorithm. And I would just, go along with Magritte, who was trying to remind us here, and this is a classic piece of art that, says this is not a pipe, that our representations of things, our formal models of things are not the thing themselves and that do not capture the entirety of the thing. And so I think this is true. And so I say this is not a Turing machine. Not even Turing machines are a Turing machine in the sense that our formal models of computation do not describe the thing, the full capacity of the thing that we are going, that we build when we make these things. And so when people argue whether computationalist views of consciousness are sufficient, my point is not only are they not sufficient for brains and living things, they're not even sufficient for the things that they were supposed to describe, which is quote unquote machines. All of these are just formal models. They capture some aspects of what's going on, but as we're seeing now, they don't capture everything. And I think again, I come back to my fundamental notion of a continuum of cognition. And it is not that you have to be somewhere on the far right of that continuum and be a complex brain in order to have some sort of freedom from being fully encompassed by models of your parts, such as the laws of chemistry and physics. That already begins at a very low level, at the level of very simple machines. Already computationalism fails there, as all formal models do. There's nothing wrong with computationalism per se. It's just we just have to remember that none of our formal models fully capture what's going on here. And so I just think it's critical that whatever their embodiment, whatever their origin story, even very simple minds are not fully defined by our models of them, neither for their limitations nor for their competencies. And so ultimately, I think the Garden of Eden is going to look like this. It's going to look like it's going to be a very weird place with all kinds of things that we are absolutely going to have to name in the sense of understanding their true inner nature, which is not easy. We're bad at it. We do not have the native skill or talent at recognizing diverse minds. We are going to need tools for it, and I give sometimes a different kind of talk where I go through this ladder where we get progressively weirder and weirder kinds of systems that I think are actually cognitive kin, and it's sort of similar to what happened in mathematics, where we progressively found stranger and stranger kinds of objects that mathematics is going to have to deal with, and each of these transitions was painful for the community. People really freaked out about irrationals and things like that. I mean, people were killed over it back in the day. And so each of these transitions across different kinds of systems, in order to understand what the level of cognition and perhaps consciousness is in any of these things, we are going to have to break all kinds of prior assumptions and prior categories, just like was done in mathematics to get to new kinds of numbers. And this is something that we're working very hard on in our group, is to try to see what do all these things have in common. And so basically, here's the summary of everything I've said today. I'm pretty confident that most useful concepts in this field are continuum models. They are not binary, crisp categories. We need models of scaling and transformation of minds and consciousness, not trying to prop up ancient categories. I'm very certain that none of this is about brains per se or embodiment in three-dimensional space. The most interesting thing about cognition and consciousness is how it explores very different spaces that we have a hard time seeing. And I would claim that our formal models do not tell the whole story, either for life or for machines or for any of these things. And because we have these cognitive patterns that show up that are not just complexity or unpredictability, they're actually new behavioral skills. And so this is stuff that we're actively working on. That is what aspects of the embodied architecture, be it engineered or evolved, matter for determining what kind of ingressions what kind of ingressions you're going to get from that Platonic space. I would claim that evolution has no monopoly on making minds, that engineering is just as potentially just as good. Of course, also because engineering can use evolutionary computation and so on. Now, Richard Watson has some arguments against this. His are probably the best. The typical assumption that this is the case, I think it doesn't hold any water. But Richard has some interesting ideas that I encourage you to check out. And we're trying to improve our tools for exploring this Platonic space and for understanding the mapping between the pointers, meaning the embodiments that we create, and the patterns that ingress through these interfaces. And the things that I really don't know what to say about at this point, I think we need to do a lot more here before we can say any of this, are questions like this. How well does consciousness actually track intelligence? I think Anil Seth's view that these are orthogonal is probably right, but I'm not sure they're completely independent. I suspect there's some relationship, but we don't know. I also don't know if there are any so-called phase transitions. I know it's very popular that to say that there are specific distinct kinds of minds, and there may well be, and the patterns in the Platonic space may in fact have distinct categories. I mean, it's certainly true for some mathematical objects like integers, but it's unclear to me whether that space is truly smooth and continuous or whether there are some discontinuities there. And what prospects do we really have while remaining more or less human for understanding very, very diverse minds? It's not clear to me how far we can get without changing ourselves, which is fine, and I think that's probably part of our future for sure. Is to change ourselves to gain a better first-person understanding of very exotic minds and be able to enlarge our own cone of concern and compassion to these other beings. So that's it. I'll stop here. If you want more details, lots of these papers here drill down into all this kind of stuff. And the most important thing is to thank the people who did most of the work that I showed you today. So these are the various postdocs and graduate students that contributed to all the results I showed you. And I want to thank many of our amazing collaborators and funding support from various agencies and foundations that have supported us over the last 30 years. And three disclosures, because there are three companies that have spun out of our work that are supporting us to some extent, and so this is part of the claim I made at the beginning that it is possible to transition some of these deep philosophical ideas into specific applications that make a difference in the practical world. And most of all, I'm grateful to the biologicals and the non-biologicals that are teaching us about these really fundamental and important questions, not only ancient questions, but I think critical for the flourishing of minds on Earth and beyond. So thank you so much. I'll stop here.